Hi, good afternoon. I'm Jonathan Cohen. I'm a Director of Engineering at Nvidia. I'm responsible for the CUDA library team and other stuff, similar kinds of things. So I'm going to talk today about machine learning, which has kind of exploded in really the last 2 years, I would say, as a very fruitful area of research, and the pace of improvement of technology is really tremendous. And so I thought it'd be an interesting talk to kind of explain to computational researchers who might not be aware of the advances coming out of machine learning community just how powerful these tools are and how you could use them in your own research. So this was a XKCD comic. I forget when it was posted, but pretty recently. So the one guy says, I want an app when I take a photo to check whether you're in a national park. And the programmer says, oh yeah, that's easy. Oh, and also, is it a picture of a bird? Oh, that's really hard. That's going to take 5 years. So this is like kind of a classic, I think, conventional wisdom, right? That vision tasks are really, really hard. Looking at a photograph and saying what it is something that I think there's been a lot of promise in machine learning and computer vision, and a lot of the techniques people have created, they kind of work, but nothing is very robust, and certainly not at kind of human level capability. And so a group at Yahoo and Flickr, in their machine learning computer vision research group, they thought this was kind of a cool guerrilla marketing challenge. And they said, you know what, actually computer vision is really good these days. And in fact, you can look at a photograph and tell whether it's a bird or not. And so they put together this website. I was going to do a live demo, but the website itself is kind of flaky, so I decided not to. But you should go there. It's just parkorbird.flickr.com. You drag images into it, and it'll tell you if it thinks it's a bird, and it's pretty good actually. And if you're geotagged in the image metadata, it'll also tell you if you're in a park, right? Which is easy. Anyway, so I think the point here was, XKCD was sort of making a joke based on this conventional wisdom, and it turns out actually the field is moving so fast that that's not really true anymore. Computers are learning to see and understand the world around us. There's a contest called Imagenet, or the Imagenet Large-Scale Visual Recognition Challenge, which has been held since 2010. And it's a pretty cool contest. It's academics, a lot of industry groups participate. And the challenge is there's 1.2 million photographs taken from the web, and they've been tagged into 1,000 categories. So here's some examples of from that data set. So this was a tremendous effort to get this data set. And the contest every year is, given, I forget the number, but given some number of images, say what's in these images. And what you see here, if I can show you here, the classification error. So they consider something correct. The computer has 5 chances to guess, and if any of those 5 is correct, then it's counted as a correct answer. And so in 2010, the 28% incorrect. In 2011, 26% incorrect. And what happened in 2012, so this is sort of typical progress in machine learning and computer vision, right? Every year, you sort of chip away 1 or 2% off of these benchmark problems. Going from 26% to 16% in 1 year is an amazing, it's an astounding result. And the team that won in 2012 used something called a deep neural network. And they trained deep neural networks on GPUs. And so they basically they said, hey, this technology really works. It needs a lot of data, and it's very computationally expensive, but it maps great to a GPU. And it turns out that we actually can make it practical by using a GPU to train. And so that team, so this is kind of our favorite chart, right? That team that year used neural nets, and they won, and they beat the pants off of everyone else. And then the next year, most of the teams were using GPUs. And then by this year, every single team except for one was using GPUs and deep neural nets. So the entire field overnight, really since 2012, totally flipped in the approach they're taking. And basically now I think deep neural nets are considered the state of the art, and everyone uses GPUs for them. So what is a deep neural net? I only have 20 minutes total for this talk, so I'm going to give a very, very high-level view, but hopefully this gives you a little intuition about what's going on. So a neural network is a very old concept. It's simply a set of transformations that are called layers, and each layer computes some kind of either linear or nonlinear function from its inputs to its outputs. So in an image recognition neural network, typically the input neurons, you would have one neuron corresponding to each pixel in the image. And you would run this through a sequence of these layers, and the output would be a set of neurons corresponding to something like, the name of the person. If you've trained it on a database of 1,000 people, you'd have 1,000 output neurons, and then that neuron would fire if it thinks it's, Sarah in this case. So kind of the history of neural networks, for various reasons, people never made them very deep. Depth in this case refers to the number of layers. And so the conventional wisdom was that you don't need more than 3 layers. And in fact, when people tried to use more than 3 layers, all the algorithms we have for training these networks basically failed. And it was really innovation starting around 2006 from Jeff Hinton and Jan LeCun, culminating kind of, I would say, in 2012, where they really sort of exploded. The idea that you actually can use these deep networks, you need a lot of training data, and you need slightly better algorithms for training them. And so what happens when you train a deep network is you find, and this is just sort of automatic in the training process, that these different layers learn to do different things. And as you go deeper through the network, basically each layer somehow creates a more abstract representation of the input. So the first layer in an image recognition neural network usually learns to detect things like edges. And so you have a set of neurons corresponding to all these different orientations of edges and basic things like corners. And then in this case, this is a network that was trained on faces. The next layer up will say, well, I'm going to look for a set of edges where there's kind of an edge like this, an edge like this, and an edge like this. And in this case of this top left, this eye detector, there's going to be a white corner next to a black corner. And so this thing, this kind of eye shape, right? You can see it's composed of these simpler primitives. And now this layer in the neural network is going to look for an eye that looks like this. The next layer in the neural network is going to look for 2 eyes next to a nose, which we've detected previously, right? And as you go deeper through this network, you're basically, somehow it's creating a more and more abstract representation of the input. And it's transforming it into a space where basic types of machine learning classification tasks are actually very efficient. And so that was really the key insight. The depth corresponds to greater abstraction, which actually corresponds to much better performance at these kind of abstract tasks like image recognition or speech recognition. There's a lot more to learn, and obviously in 2 minutes I can't say too much, but it's a really rich field and it's moving extremely fast. So what is the point of machine learning, or how is machine learning different from more traditional programming? So as a programmer, if you say, I want to write a program to detect a bird in an image, you would think about what's the algorithm, right? I'm going to look for a beak, I'm going to look for wings, whatever it is. The beauty of machine learning is that you don't write a program, you just feed data, and somehow the program writes itself in the form of weights in these neural networks. So the process is like this. We have a large corpus of training data, labeled in this case. So I have a picture of a dog. I run it through my neural network. So I start out where all of these weights in this neural network, all these little oriented edge filters that I showed you, they don't start out like that. They just start out as random noise, basically. I run the dog through this random network, and it says at the end, I think that's a turtle, because it's random. And basically we say, no, it's not a turtle, it's a dog. And so what we do is we basically push all of the weights a little bit in the direction that would tend to make it say dog instead of turtle. Right? This is a gradient descent algorithm, if you're familiar with that. And these weights propagate backwards through the network in an algorithm called backpropagation, which is really just the chain rule for the gradient. And so we've now updated the network to be a little bit better at detecting when it sees a picture of a dog. And now we showed a picture of a cat. And we show a picture of a tree. And you show it 1 million images, and you do it many, many times and let this run for a few weeks. And at the end of this, you have a set of weights in this network that you've trained, which you can now deploy, right? I basically have a program I can now execute. And I deploy it by showing it a picture of something else. This is my cat. And it looks at this thing and it says, oh, that's a cat, or so you hope. And so that's the basic idea, that you're sort of trading programmer expertise for lots and lots of data. So that's kind of a quick overview of deep learning and how it works and why it's so exciting. What I wanted to talk about today was just highlight a few applications that I'm aware of where people are actually using this technology to improve science. And I think as a computational scientist, one thing that's sort of interesting to think about is there's a new tool that exists now, which is something that can look at an abstract image or more research topic, video and audio signals, and actually make sense of it. And so you could ask the question, how could you make use of that tool in your research? And so here are 3 really great examples of people who are doing that. So the first is a coral reef mapping project. Coral reefs are, so my brief disclaimer, I'm not a scientist in any of these domains, so I'm just saying what little I know here. Coral reefs are tremendously important in marine ecology. They support more species per area than any other marine environment. And they're a storehouse of immense biodiversity, which is important for all sorts of reasons. Healthy ecosystems, drug discovery, sort of just generally understanding nature. They also have a function where they buffer adjacent shorelines from wave action, which is very important, obviously, to prevent erosion. Coral reefs are full of this biodiversity, and it's very important for people who study them to have accurate large-scale maps of coverage broken down by genus, for instance. So this is actually an example image that Oscar Baybaum sent me, showing the kind of coverage maps you would see. So this is Australia, Great Barrier Reef, and you zero in on the Wilson Reef, and then this is showing a map of total coral coverage and then different types of material in the coral reef with these density maps. So how do you generate a plot like this? Well, they do these enormous surveys, and they have autonomous underwater vehicles. I think the NOAA runs these kinds of survey projects. They're kind of large-scale, expensive efforts. They have these towed things that go behind boats and take lots of pictures. And so you're able to generate now, or the survey organizations that do this are able to generate enormous image data sets, which are good. But at the end of the day, you'd like to turn that into a map, which means you have to label it. And labeling is tedious and slow. And so anecdotally, only 1% to 2% of all image data obtained from these surveys is actually labeled. And this is actually an example of what this labeling looks like. You can see how incredibly time-consuming this would be. So in the circles are showing different coral genera. These are some types of coral. And you can see they're actually annotated over the photograph, right? This is that, this is that, this is that. And then triangles are the non-coral substrate, things like sand or algae. Right? And again, these, imagine as an expert sitting down and hand labeling this, and then you get the next one, you hand label it, and the next one, you hand label it. This is a very tedious, error-prone process. So there was a challenge problem to replace hand annotation with some kind of automatic annotation. And the best methods today work on about 60% of the data. So if you replace 60% of the hand-labeled data with automatically labeled data, you get about a 5% loss of accuracy, which is deemed acceptable, or perhaps deemed acceptable. There's a pilot project underway, so this is actually proposed research. They don't actually have results yet. Where they believe with deep learning, they can actually raise this to about 90% of the data could be processed automatically with this 5% loss of accuracy. There's a lot more information here. Because they don't have results, I still thought this was just a great example of how this is such a cool tool for research. And the core technology here is, again, deep neural networks running on GPUs. The Connectome project is an amazing sort of grand challenge effort, which is mapping the wiring diagram of the brain. There's a quote from the Harvard Center for Brain Sciences. Imaging neural circuits at nanometer length scales leads inevitably to vast data sets. So think about that, right? You have a three-dimensional brain. The scale that you need to look at is on the order of nanometers. So these are just tremendously large data sets. In fact, the entire Connectome project is feasible only now because of the exponential increase in computing power and data storage over time, which is absolutely true. So how does this work? So they take these samples of brain tissue. This is a mouse brain project from Harvard Center for Brain Sciences. They basically shave it into these tiny little slivers, put it in an electron microscope so that you get these images. But this is just a bunch of pixels, right? You have to make sense of it. So first you have to determine where are the membranes, and then you have to segment this into different neurons. Do this for every single slice that you've generated, and then convert this into a three-dimensional image. There's actually some examples over there of what the output of that sort of thing looks like. The microscope they have at Harvard is generating just under a terabyte of data per day right now. They have a cluster of 250 Tesla K40s just running the classifier. So this is, again, a deep neural network that's trying to classify membrane versus non-membrane. The next generation microscope, which they're bringing online now, will generate a gigabyte of data per second. and they plan to run it about 50% of the time. So that's 42 terabytes of data per day. Again, hand annotating this is just impossible, right? And I wanted to show you a short video. This is from Jeff Lichtman's lab at Harvard, showing the reconstruction of just a single little cylinder of this data. This is all the neurites, which are the axons and dendrites, of one little cylinder, 15 microns long. So this is a tiny cylinder. And you can see all these little things. These are all important, right? And they did this really amazing visualization. I think this is part of a Discovery Channel program. You can see it on YouTube. And so I asked, what was the how was this reconstructed? So this was actually done by hand by 2 postdocs working for about 9 months, which illustrates just how important it is to automate that process, right? That's a tiny, tiny little piece of this data set. 9 months of 2 postdocs. And then the last project I'm going to talk about is cancer cell mitosis detection. So mitosis is the process where the chromosomes in a cell nucleus are replicated prior to the cell itself splitting. So when you have cancer cells, right, they replicate very fast. And so there are different grading systems people, pathologists use to understand whether tissue is cancerous or not. And one of these grading systems uses basically a count of mitotic activity per tissue area. This is the BNR grading system. I think it's one of the 3 criteria used to determine whether something is cancerous and how cancerous it is, or how invasive it is. And so it turns out that this count of mitotic activity is a strong indicator of invasive breast cancer. And so the way that this is done is, again, you take a tissue sample, you stain it, you put it on a little slide, and a pathologist looks through a microscope by hand and labels the cells that are undergoing mitosis. And so this I just grabbed from the website describing this particular challenge problem. Shows what you're looking for. Maybe it's a little hard to see, but this green arrow is pointing to a cell that's undergoing mitosis, and this green arrow is a cell undergoing mitosis. And so imagine how, again, painstaking and laborious and monotonous it is to look at slide after slide after slide and hand label this over a 2 square millimeter area of tissue sample. But this is what's necessary for understanding the tissue samples that may or may not be cancerous. You can also see this is actually a very difficult visual recognition problem, right? That, you know, our brains are really good at this, but there's just a lot of stuff going on in this image, and understanding, oh, these things are the chromosomes, and here they're undergoing mitosis, and this is what's happening. It requires a certain level of expertise. And so there, for 2 years, there have been contests from a medical imaging conference where they basically created these hand-labeled data sets, and then they fed them to computer vision researchers and said, you know, can you automate this process? And so a group from Idzia, led by Dan Cherishan, won this contest for 2 years in a row. Again, using deep neural networks, and in fact, he was really one of the pioneers in applying deep neural networks to these problems of medical imaging. So the idea here is that the deep neural net is a pixel classifier. So the input is a window of raw pixels, and then the output is probability that the center pixel is close to some mitosis centroid. And so you slide this window over the image, and wherever you get your strong hits is where you expect to see some mitosis. They have these pretty large data sets. So in 2012, it was 66,000 positive examples, 6 million negative. Running on a CPU would take 5 months to train this neural network. On a GPU, because they're really good at this, it took 3 days. I think that's just a really important part of the story, right? GPUs and the software that's been developed to run on them is one of the reasons why this actually is happening now. Because 3 days is tractable and 5 months is basically not. And they won. So this is an example, or this is one of the plots from their paper showing the accuracy of their results. This is an F1 score, which is a harmonic average of recall and precision. So some kind of measure. There's, you know, the accuracy is actually kind of a whole curve here. So this tries to boil it down into one number. And again, you can see they really won very solidly versus the other entries. Dan's giving a talk immediately after mine. I encourage you all to stick around and hear what he has to say. He's going to go into this in a lot more detail. So how do you get started? And I'll wrap up in 1 minute here. So anyone can use deep learning. It's still a research topic for sure, but some of these basic technologies of learning image classifiers are actually pretty easy to use. There's many great open source frameworks available, and they all have active user and developer communities. Caffe is from Berkeley, Torch is from NYU and is heavily used by Facebook. Theano is from University of Montreal, which is a Python, integrates very nicely with NumPy and SciPy. So take your pick based on your preferences. All of them have excellent online tutorials. Here's some links to them. Check them out. And every single one of them runs great on Nvidia GPUs. Buy the biggest, baddest gaming Titan you can, put it in your machine, or put a bunch of them in your machine and get to work. And I believe, and I think scientists who've been experimenting with this that I've talked to believe that this is going to impact just many areas of science: astronomy, sociology, political science. You want to look at photographs taken at political events and categorize, what are the races of the people who showed up, and that, all sorts of things you can do when you can analyze these images. And just the last plug, so my team developed a library called QDNN, which actually plugs into each of these frameworks and accelerates all of them. So it's a library that has high performance routines for a particular kind of neural network called the convolutional neural network, which is really the popular type these days. It's optimized for NVIDIA GPUs. You can use it directly if you want to hand code your own, but if you want to use one of these frameworks, you basically just drop it in and things get faster. So this is showing a particular benchmark, Caffe before and then Caffe using QDNN. They just got a boost. And the next version we're going to release pretty soon will give you an even bigger boost. And so I think with that, I'll stop and take any questions.